So, last time we talked about female reproduction, we talked about oogenesis, we talked about the process or like the menstrual cycle. So that kind of 28-day hormone cycle where we have specific events occur in the process of both oogenesis, ovulation, how we also spent a lot of time talking about meiosis, like talking about how we take our lots and lots and lots of DNA and to subdivide it down into much smaller portions. By much smaller, I mean by half. But where we're making sure that we're able to move exactly one of each chromosome into each gamete so that we can move basically one half of our genetic information onto the next generation. Okay. Well, the female reproductive system is trying to accomplish a couple of things. One, it's trying to, it needs to release one, maybe two oocytes every month. And then it's trying to basically transmit sperm cells from the male and get them to those oocytes. And then if those oocytes are fertilized, it then needs to be able to grow and nourish that growing embryo, and then eventually be able to expel that growing embryo, or at this point a baby, say 40 something odd weeks down the line. Okay. That's not straightforward. That's a lot of stuff happening. However, the process of making that single gamete can be, well, at least somewhat straightforward, because like you're trying to do that once. Okay. And the pathway that that gamete is moving is actually fairly short. It's going simply from the ovary into that, through those fimbriae, the kind of finger-like appendages, through the infundibulum, which is that kind of hand-shaped region of the fallopian tubes, and then finally into the uterine tubes, and then taking it down to the uterus, right? And then we have the female copulatory organ, which is the vagina, which is basically going to be both the birth canal as well as the passageway through which sperm and stuff like that will travel from the male. Now what we're going to talk about is the males. Now, the males, because they are not having to grow and develop the growing embryo, instead can put all of their effort and energy into making and delivering as much genetic material as is possible. Okay. Now, because of that, we have a few things that we now have to deal with. One of these is that the production of the male gametes, production of sperm, is actually needs to happen a couple degrees cooler than body temp. Okay. When temperatures get too high, some of the proteins and stuff that we are using are going to break down, and the sperm will not be effective, and can be completely or made completely ineffective just simply because temperatures are too high. So temperature control here matters. So because of that, we have male organs, unlike female organs, being found outside the body. Okay. So that is why the testes and things like that are found outside the body. The penis being found outside the body makes a lot of sense just simply by looking at the genitalia of the females, or looking at the vagina, noticeably that it is interior, which means on the male end we have to be exterior. Okay. Now there are some similarities here, though. We spoke about the clitoris last time and how the clitoris is going to be both external and internal, and it's going to be composed of this sort of spongy erectile tissue that can hold on to blood, which is going to make it be somewhat firm. The same thing is going to be said for the penis, which is basically just going to be essentially like a very large clitoris that happens to have a urethra passing through it. Okay. In many ways, they are very, very, very similar. Okay. Even that the penis has a lot of that erectile tissue being found inside. Okay. So being found deep, or at least anterior to the rest of the outlying tissue. Okay. Now, our pathway here is going to be fairly straightforward. We're going to produce sperm down here in the testes. It will be stored here in the epididymis. This is also where it will kind of mature. That's to say, get rid of everything that is not genetic material, acrosome, which is this little cap on the end of it, and then all the mitochondria that are needed to power its single flagella. Okay. That process is going to happen here in the testes, and finally in the epididymis, and then it will be stored here, and then finally it will be expelled out through the structure here, which is called the ductus deferens, or the vas deferens. Okay. So it will get pushed up through this vas deferens, where it wraps around anteriorly. It's going to go pass over the pubic bone. And then it will kind of wrap around to the backhand side of the urinary bladder, at which point it will come into contact with this other structure here, which is the seminal vesicle, okay? Now, you may have heard about the male ejaculate being called semen. Well, there's a lot of products that are going to be found in the male ejaculate. It's not going to be just sperm. It is going to contain multiple fluids, okay? One of those fluids is going to come here from our seminal vesicles. This is where we're actually going to get our seminal fluid. And this is going to be where we're going to get the bulk of the fluid that is going to be surrounding the actual sperm, which is coming from, once again, the testes, okay? So the vast majority of the fluid that is actually going to be ejaculated from the male will actually come from this gland right here, which is our seminal vesicle, okay? Now, after the vas deferens comes into contact with the seminal vesicle, we're now going to be in what's called the ejaculatory duct. Now, outside the ejaculatory duct is going to be this big kind of walnut-shaped structure here, which is called the prostate gland. This is something that is, once again, also only found in males, okay? You'll also notice that it is also wrapped around what we call the prostatic urethra. So we have the urethra passing from the urinary bladder, and it will pass through the prostate in this region, like I said, called the prostatic urethra. So we have the ejaculatory duct and we have the prostatic urethra. The prostate gland is going to be secreting its proteins into this urethra, into this ejaculatory duct, where then those will combine, and then it will pass into the internal and finally the external urethra. We have one other gland that we're going to be passing by though, and that is our bulbourethral gland. Okay. Now we're going to talk about the substances that each of these is secreting, but simply based off of size, you would probably imagine that the bulbourethral gland is going to be the smallest. And that would be correct. However, this is also going to be one that is going to act as kind of a primer for the urethra, because once again, the urethra is also going to be used for urine. And we know that urine typically has a fairly acidic pH. That acidic pH could actually be enough to kill some of the sperm here. So we don't want that. So prior to the ejaculation, with all of the sperm and all the seminal fluid and stuff like that coming through it, we do actually need to kind of prime the urethra for the sperm to come through by getting rid of some of that acidity. So we need to have some alkaline products here just to make sure that we can balance out the acidity that's there, okay? Now, we can also see the proximity of all of these structures to both the rectum and anus back here, as well as the pubic bone more anteriorly up here, okay? Now, all of these external structures, or at least the testes, epididymis, and ductus deferens or vas deferens, these are also going to be surrounded by a couple layers of muscle, okay? Namely, we have a layer of muscle here that is going to be deep, covering the testis and finally the spermatic cord. And then we're going to have another layer of muscle, which is basically forming the floor and kind of the outside of the scrotum before it is covered by kind of this external bit of fascia. Okay. Now, these muscles here are going to be responsible for allowing the testes to be moved closer together and closer to the body, or further apart and further from the body. Okay. The dartos muscle, because it is going to be found here outside, this one is going to be the one that's actually going to pull the testes closer together as they have been tucked up closer to the body. Basically, by holding them closer together, it's kind of like whenever you pull your arms and stuff in, whenever you're cold, it's basically pulling everything in together to keep it nice and warm. The cremaster muscle, which is going to be this muscle that's surrounding the testes and its spermatic cord, these muscles are actually going to pull the testes up, okay? So cremaster muscle pulls it up, dartos muscle pulls them in. All of this in conjunction is going to pull them up and in and get them closer to the body trunk in conditions when it's cold. When conditions are warm, these muscles can relax and allow the testes to move further away from the body so that they can stay nice and cool, okay? Now, also to help them stay cool is going to be this really cool kind of counter current of blood supply here. Now, because the blood is warm up here, and it is moving down through the tissues, we want to cool that stuff down as much as we can. Well, like I said, we're going to be kind of moving the testes and stuff like that away from the body. The further it is away, the less of that temperature is going to get, which means some of that temperature is going to get lost. So the venous blood that is returning from the testes is already going to be cooled or cooler than the arterial blood that's coming down. So we get this cool structure here called this pampiniform plexus. That's this blue kind of, let me see if I can blow it up. Oh no, that's actually exactly what I was supposed to do. Because I'm not sure how easy that was to see. So now that's a little bit easier to see. And it's also easier for me to see, so I can talk about it. So you see this blue network of veins, okay? By opening these veins up, rather than just being a single venous return, when it's a single artery in, we have this multiple sets of venous return kind of fanning out. This is going to both allow that blood to get cooled because it is close to the surface, but it's also going to be acting as a heat sink for the blood that is coming through the artery. So because we have this warm blood coming through the artery, and it's going to be wrapped in all of these cold veins around it, these cold veins are going to pull heat from that artery and pull that heat out. What do you need? Here's 2.
Thank you.
So warm blood is coming through, but it's got cool blood around it. So what does heat do? It's going to move from high concentration to low concentration. So it's going to move from that testicular artery into the vein. That both cools the blood even more as it's on the way to the testes. So the testes aren't getting too warm because they're getting hot blood from the torso. They're also so we're cooling the blood as it's coming down, but it's also warming up that venous return because that blood is coming from somewhere where it's cool. That blood is now cool. So we're kind of warming it up a little bit so that we're not kind of cold shocking any of the testicular veins up here. That way they're not super cold, so they're not going to be pulling heat away from our other underlying tissues and things like that. So we're basically able to kind of swap the heat back and forth to make sure that blood that is re-entering the body is going to get a little bit warmer, and blood that is going down to the testes is going to get a little bit cooler. That way, once again, we can maximize the sperm production and the health of the sperm. Okay, so like I said, that is our pampiniform plexus. And all of that is going to be found inside what we call this spermatic cord. So that's going to be the muscle, going to be the testicular artery, the pampiniform plexus, which is that vein, the ductus deferens, as well as the testicular nerve, which is going to be this big nerve down here, which will innervate all of the structures down here in the testes, as well as these muscles down here. Now, once we get inside the testes, we're going to see some structures that are going to look very, very different than what we saw in the ovaries. In the ovaries, our goal was to produce a single mature secondary oocyte. We want that cell to be big, happy, healthy, and perfect. Okay? Not the case in sperm. Yes, we want these things to be as perfect as we can. However, there is very much a sort of shotgun approach when it comes to the sperm production, in that we are trying to make as many as we possibly can as fast as we can. Okay. So because of that, we now have the same structures that same type of structures that we've seen throughout the body. Whenever we need to do a whole lot of something, what do we need to do? We need to fold it. We put more folds and more folds and more folds and more folds so that we can pack more stuff into a small space. Okay. That's what we get here in the testes. We're going to get a whole lot of these little folds and curves and stuff like that so that we can fit more of these little tubes inside the testes. These little tubes are what we're going to be calling the seminiferous tubules. And these are actually where we are going to be making sperm. Okay. Now, the testes themselves are going to be separated into like these little lobules. Okay. With their own layers of, whether it's visceral parietal layers of the tunics. So these tissue tunics kind of surrounding them, but we're still dividing them into their own individual lobes, where each individual lobe will then make its output and put its output here into this region in the middle, which we're going to call the mediastinum testis. That should sound very familiar because that's basically the same root that we had for the mediastinum in the chest. This region is also sometimes, or will contain the structures called the rete testis. That's basically where the individual seminiferous tubules from each lobe will come together in this kind of rete testis. And then those structures, or those sperm, will then get migrated out here into the epididymis, where they are stored until they are used. Okay. Now, inside the seminiferous tubules, we're going to see something like this. So here we have a cross section of one of those seminiferous tubules, keeping in mind that we have thousands of these. Okay. You'll notice that we have things that look like sperm down here, and they are sperm. They are just not quite mature. They're not quite ready to go. They still need to make some changes to their little caps and stuff like that. So we have immature sperm here, but they are sperm. We have genetic material with a flagella inside the lumen of the tube. These just need to get moved to the rete testis, where they finish maturing, and then get moved out to the epididymis, where they are stored. Okay. So we have sperm here in the middle. So that means likely out here around the outside, we have essentially protosperm. And that's exactly what we're looking at here. Okay. Around the outside of these, we're going to start with what we call spermatogonia. Okay. So these spermatogonia are going to be the parent cells for the things that will become our sperm. Now, because these are our sperm, that means these are the end result of meiosis 2. These cells are at the start of meiosis 1, so in the interphase before meiosis 1, or maybe even prophase of meiosis 1. What's that mean? Well, that means that we need to go through meiosis 1, which is that first division. So we need to double our DNA and divide, which we've done here. We then need to line that DNA up in the middle of the cell, divide it into daughter cells, and then those cells then need to divide out here. As we finish that second division, we will now be all the way out here. We will grow the tails as we are in this last little stage out here. These spermatids out here, then we grow them as tails, and then finally they are sperm. Now, intermixed in here, we will have these sustentacular cells. The sustentacular cells are important because these are going to be secreting. Oh, you're fine. What do you need? We've got these sustentacular cells, which are going to be the ones that are actually making our testosterone. So like I said, we're going to start with our spermatogonia on the outside. These will then be dividing and pushing daughter cells out towards the center of the cell, and out towards the lumen of the seminiferous tubule, at which point they will be sperm. So at any point in here, you'll have cells. The closer you are to the outside, your cells will be either in meiosis 1, further away from the outside, closer to the lumen, they'll be in meiosis 2, essentially undergoing that double set of divisions. Now, all of this is going to be regulated by gonadotropin-releasing hormone, okay, which is in our hypothalamus. That should sound familiar because that's exactly how it started in the female reproductive system. Gonadotropin-releasing hormone, just like it did in the female reproductive system, releases follicle-stimulating hormone and luteinizing hormone, LH. I don't know about you, but in this picture, I don't see any corpus luteum. I don't see corpus luteum here. So what is the LH doing? Okay. Sorry, I said the wrong thing a second ago. The LH is actually going to be stimulating our interstitial cells. That's going to be these little guys out here. So interstitial meaning between the tubes, so inside the tissues. So here's a seminiferous tubule, here's a seminiferous tubule, here's a seminiferous tubule here. So these interstitial cells are going to be the cells that are in between each of these. These are going to be secreting testosterone. The secretion of testosterone is going to cause additional secretion or additional sperm production. So they're going to tell those spermatogonia that they need to divide more, okay? Now, follicle stimulating hormone is going to stimulate our sustentacular cells to secrete this androgen binding protein. Androgen binding protein binds testosterone. So we make testosterone in the interstitial cells between the seminiferous tubules. Then after we've made it outside the seminiferous tubule, it will kind of work its way in and get bound by this androgen binding hormone. And this is going to make sure that we're maintaining these high levels in the testes so that we can maintain this high level of spermatogenesis, okay? Now, higher testosterone levels means we're going to get this additional bit of spermatogenesis. So we'll be making more sperm as kind of a byproduct, okay? Now, this will also, just like we saw in the female reproductive system, this will act as a negative feedback mechanism. high levels of testosterone will downregulate or will cause the downregulation of gonadotropin releasing hormone, which will in turn downregulate the release of luteinizing hormone and follicle stimulating hormone, which would then turn off sperm production. So what does this mean? Well, this means that if you are utilizing the sperm that you're making and you are utilizing all the testosterone that you're making, that is facilitating that spermatogenesis. So you're making a whole bunch of sperm, but that sperm is being used. You will continue to have this kind of high level of testosterone. However, if say you have a backup of sperm, so you're not utilizing the sperm that is being stored in the epididymis, well, you don't need to be producing more sperm. So what that's going to do is it's basically going to send a signal to the hypothalamus that we don't need to make additional sperm. We don't need more help, which means we also don't need more testosterone. So it will downregulate the sperm production and the testosterone production that is caused by follicle stimulating hormone and luteinizing hormone. Okay. At the same time, those sustentacular cells that we were looking at before, these are going to respond to rising sperm count levels. So if you're having a rising sperm count, once again, you're making a whole bunch of sperm, well, if you're making a whole bunch of sperm and you're like, hey, this is likely a good amount of sperm that is being produced right now in this time, the sustentacular cells release inhibin, which does the same thing. It causes the inhibition of FSH secretion. So we are not inhibiting gonadotropin releasing hormone. Instead, we're inhibiting the next step. So in this case, we're inhibiting follicle stimulating hormone, which means, once again, we have this other thing that is causing the downtick of the release of, in this case, follicle stimulating hormone, which was testosterone. Now, did I skip a slide? I guess I did. Testosterone is also going to play a role in both sexual maturation of particular characteristics, whether it's the growth of body hair, deepening of the voice, growth of facial hair, body hair, things like that, as well as things like libido and the other secondary sexual characteristics. Okay. Now, if you take someone that is already fully developed, okay, so say you have someone that is already a full-grown adult and you basically drastically uptick their testosterone levels, this will highly uptick their libido, while at the same time, it can lead to deepening of the voice because it will actually can change some of the growth characteristics or continuing growth characteristics in places like their nose, their ears, their cheeks, sometimes even their jawbones, which can lead to a deeper voice. Their hair will also kind of uptick. So whether it's the growth of their actual hair, facial hair, or pubic and axillary hair, all that stuff can uptick. However, it can also cause, like say they are doing this with like just drugs that they're taking, like say they're bodybuilder types or something like that, that can also cause a slowdown or at least a decrease in the size of some of these testicular structures, which are responsible for doing this stuff down the road. Because like, hey, we don't need to be producing as much sperm, or we don't need to be producing all this testosterone, which leads to these downstream effects of lots of sperm production and stuff like that, because we have all this other testosterone that's being added to the system. So because that, you can actually sometimes see individuals that are undergoing those types of treatments, you'll see them have reduced testicular size and stuff like that because their testes are essentially regressing because they are not using their testes for what their testes are also for in the case of the hormones. Oftentimes, you'll also see this be accompanied by them having lots of acne and stuff like that, which are some of the other characteristics that can come from increased testosterone, which is even one of the reasons why you tend to see when testosterone levels start to pick up in adolescence. So like young teenagers and stuff like that, whenever they go through puberty, their bodies kick in and start producing testosterone, their bodies aren't used to that. Their body chemistry isn't used to that. So that's when they'll start to get lots of pimples and acne and stuff like that too. So like I said, we start with our spermatogonium out here. These have our double DNA. So we've got our normal levels of DNA. So we have 46 total chromosomes. We have 23 pairs of chromosomes. These will then divide into what we call the primary spermatocytes. Where one of these basically say the spermatogonium, one becomes primary spermatocyte, then they will continue dividing. These will undergo meiosis. Now, our first set of meiosis, remember, is going to give us two sets of our chromosomes where we have that one paternal or one familial copy and then the one blended copy that is attached to it. Then those will divide again, where that's when we'll separate those into their individual daughter cells via, where'd my pointer go? Via meiosis II. And then these are what we're going to call spermatids. So until they have grown their tails, they will be spermatids. Once they grow their tails and you start to see this regression of cytoplasm and things like that, you're basically being left with nothing but a flagellum and a whole bunch of mitochondria that power it. That is when they will now be kind of our sperm. Okay. So we get rid of all that excess cytoplasm because we don't need it. We get rid of all these extra structures because we don't need them. We're going to be left with nothing but this acrosome cap, which is basically this tough protein cap that also has signaling molecules that will attach to some of the those kind of helper cells on the outside of our ovulated secondary oocytes so that they can bind and say, hey, that's a sperm cell. That means we can let that thing in. And then we're going to be left with the nucleus behind it, which contains all that genetic information, a whole bunch of mitochondria, which are going to power this tail, which will then swim and swim and swim and swim and swim until it finds the egg. Okay, so to compare these two things, female gametogenesis, we produce one single viable secondary oocyte. All three of the other copies are what we're going to call bar or polar bodies. Okay, so these things are not used. We still produce them, but they are not used. The first division at meiosis I, that one that gets made just kind of gets dropped off. Okay, the second one, so then we're left with that single primary oocyte or secondary oocyte that hasn't finished dividing. If it is fertilized by a sperm, it finishes its second division. It drops off 1/2 of that DNA as another polar body, at which point we then now have a viable embryo. In males, we produce 4 sperm. It's that simple, that straightforward. We start with 1 cell, we produce 4 sperm, and it's basically done at that point. No bar bodies or anything like that. Any cell that gets made gets used. And one of the reasons that this will happen, because remember that one of the things that happened in the production of our secondary oocyte, or choosing the secondary oocyte that was going to be the one that donated its genetic material, is that we have this uneven distribution of cytoplasm. We have one cell that gets the vast majority of the cytoplasm of the division, and then we have this other cell which gets this small proportion. Because you get this small proportion, that cell's no longer viable, that goes away. Well, in sperm cells, we don't need a whole lot of cytoplasm. We just need enough cytoplasm that we can produce all the mitochondria and hold all the mitochondria that we need around that flagellum. Because of that, because we're getting rid of the excess cytoplasm anyway, not having perfect division of, not having perfect cytokinesis, so this perfect division of the cytoplasm, isn't really a problem. Also, female oocytes are arrested in meiosis prior to birth. Remember that they are kind of halted in the beginning of meiosis I. At birth, you start with all of the eggs in a female that person will ever produce. You can't make them produce more. Males don't have any sex cells at birth. Instead, they start that process when they hit puberty. Females can only reproduce until menopause because at that point we stop ovulating those secondary oocytes. Males will continue to produce spermatocytes or will continue to produce sperms and spermatocytes, primary spermatids, all those fun things throughout the entire lifespan. However, over time, they will accrue some kind of mutation debt, essentially. Over time, they will accrue enough mutations that you will see an uptick in birth disorders and stuff like that associated with the offspring from males that are late in their life, just simply because they will have accrued a lot of problems. All right. So here we have a different view. This is a posterior view. We know this is a posterior view because we can see the urinary bladder here and we can see the seminal vesicles. Seminal vesicles, remember, are going to be found on the posterior side of the urinary bladder. We can also see the ureters kind of passing down to the posterior side of the urinary bladder. So we know that this is posterior. Now, we have the testes down here. We have the testicular lobules. We have the rete testis, and then finally we have the epididymis, and then we're going to go up through the ductus deferens. Okay, up through the ductus deferens, up through the ductus deferens. Notice that we do have a muscularis structure here, with a fairly small lumen inside that is actually going to be used to propel the sperm forward. Basically, it's going to be like squeezing toothpaste through a tube. So it will essentially just use like peristaltic style contractions to push the sperm up through the ductus deferens past the seminal vesicles. Now, once we get to the seminal vesicles, now there's enough fluid where the sperm can start to swim. They're not having to swim this entire distance, okay? Once we get enough fluid around them, though, they can actually start to swim. You can kind of picture this like if you see a swimming pool. 5 people swimming in a lap pool, not that big of a deal. There's enough room where they can all kind of get out and do their own thing. If you put 5,000 people in a lap pool, it's going to be very, very difficult for the people that are in there to be able to swim effectively because they're just going to be bumping into everybody all the time, okay? So the sperm cells need a bunch of fluid just so that they actually have enough freedom of movement for the flagella to actually move them through the liquid. So that's one of the reasons why we have to have this kind of liquid basis for the sperm to be found, and we can't just have this like sperm paste be used for this style of sexual reproduction. And when I say sperm paste, there are actually some animals that don't do sexual reproduction with sperm this same way. Some of them will put like sperm in like a little packet that the female will grab and kind of then touch to her area where she then releases her eggs, like touch to her cloaca where she releases the eggs through those. And that's actually how we have these sperm cells meet the egg cells and stuff like that. I'm thinking about salamanders in general. When I say this way, spiders do it even differently. Although they also use like little sperm packets. In the case of spiders and scorpions, they can hold on to sperm packets for years before they use them. It's all very bizarre. Not bizarre, it's just very different than humans, but kind of cool. All right, so once again, we have the ejaculatory duct, which is where we have the connection between the seminal vesicle and the vas deferens. And then we have the prostate gland, and then we have the prostatic urethra, at which point we now have the prostaglandins and things, or we have the products of the prostate gland that are secreted into the urethra. Then we pass into the urethra, passing by our bulbourethral glands, which are secreting their own substances. In this case, I believe those are the prostaglandins. And then we get down here into the urethra. We have 2 types of erectile tissue. In the penis, we have what we call the corpus spongiosum, which is going to be the inner layer, which is around the urethra. So we call this region of the urethra the spongy urethra because it's passing through the corpus spongiosum here. And then we have this corpus cavernosum on the outside. So the corpus cavernosum and spongiosum are a little different because the corpus cavernosum is going to be composed of like these little pockets of erectile tissue on the outside, which are going to be at a slightly different angle to this kind of just overall spongy texture around the urethra. So because of that, they're going to fill with blood and maintain rigidity and integrity differently than those that are around the urethra. So they are going to look a little different. All right, so now let's go ahead and talk about the stuff that's being secreted by each of those glands. So seminal fluid, first and foremost, like I said, needs to be alkaline so that we can neutralize both the acidity of the male urethra, but also we need to do this because we need to neutralize vaginal acidity. Now, because the vagina is open to the exterior environment and is really only protected by a couple of flaps of skin, it needs to be protected badly, okay? So because of that, it will actually have a mucus secretion constantly being produced there, which is going to make the vagina somewhat acidic, okay? The males need to neutralize that so that the acidity inside the vagina does not kill the sperm. We also need to give nutrients to those sperm that are trying to pass through them because they don't have a lot of cytoplasm. These things aren't able to do a whole lot. They have a whole bunch of mitochondria and a big protein that is going to be burning energy very, very, very quickly, which means they need to have nutrients that are going to help them do this, vitamin C, sugars, and stuff like that, okay? Now, seminal fluid is going to be produced by 3 different glands. We have the seminal vesicles, the prostate gland, and bulbourethral glands, okay? Now, seminal vesicles, like I said, were going to be the structures found on the posterior side of the bladder, where they will then come into contact with the ductus deferens, okay? This is going to secrete our alkaline fluid. So most of the fluid, it is alkaline, once again, so that we can neutralize that vaginal acidity. But it will also have fructose and prostaglandins. Fructose. Well, fructose is fruit sugar, okay? Fruit sugar. This is going to be important because it's going to nourish the sperm, okay? This is a sugar, easily broken down, converted into ATP by the mitochondria in those cells. So now we can nourish the sperm so that they have enough energy to continue their swim, okay? Then we've got prostaglandins. The prostaglandins here, you would think they were from the prostate gland. They're not. That can get a little confusing. So our prostaglandins here in our seminal vesicles are going to essentially be muscle relaxers, okay? As muscle relaxers, whenever this fluid touches the cervix of the uterus, remember, because that was that kind of closed down region at the opening or at the end of the uterus, the inferior portion of the uterus facing the vagina, it's going to touch that cervix and it's actually going to cause them to relax a little bit, which is going to allow the sperm to pass through, okay? Now, the prostate gland is going to secrete citric acid, and then the structure is called seminal plasmin, as well as prostate-specific antigen. Okay, citric acid is important, so this is vitamin C. This is basically going to make the sperm more efficient at making use of the fructose that is found in the fluid from the seminal vesicles. Okay, we have seminal plasmin. This is basically going to be an antibiotic. Okay, because we are facing the outside of the body, we want to make sure that any bacteria that may be in there, both in the male urinary tract or in the female, that we are having a way of combating that, okay? Making sure that we're not causing or dealing with urinary tract infections, or basically bacteria that could be attacking the sperm, making them have issues, okay? Then we've got this prostate-specific antigen, okay? This prostate-specific antigen is going to be something that is going to help make sure that the semen that we're producing is going to be liquefied, okay? Basically, it's going to make sure that we're holding on to enough water around particular structures to make sure that, hey, things are able to divide and separate and be nice and liquid as they are moving through, rather than basically just be in big kind of chunks and globs, which would make it not very effective for the sperm that are trying to pass through it. Now, this gets us lastly to our bulbar urethral glands. Here, we're looking at only 5% of total ejaculate volume here, so something very, very small. But these are basically just going to be this clear liquid that is forming a mucus. This one is going to coat and lubricate the urethra during intercourse. This is that basically by covering with this very thin layer of mucus, we're protecting the sperm that are going to be passing through from any of the acidity or anything like that, any underlying urea that may be still found in the urethra. We're able to protect the sperm from those other secretions, okay? Now, with all these liquids, you add in the sperm, now we have our semen, okay? Once it is released, we can call it the ejaculate, but before that is going to be the semen. And in each one of these, you're going to contain anywhere from 2 to 500 million spermatozoa. That's a lot of cells. Keep in mind, females release one of these every month from one ovary. You might do this twice, but only one ovary kind of releases one. Males, 2 to 500 million every time, okay? That's a lot. Now, if someone is particularly active, and this is happening multiple times, say over the course of a day for multiple times, well, if that is happening a lot, this number, of course, will go down because you will probably be releasing sperm that have not matured yet, or also just can't quite make the cells quite that fast. But in a normal ejaculate from someone of a normal kind of level of sexual activity, we're looking at anywhere from 2 to 500 million. That is a lot, okay? Now, it takes about 2 weeks in order for us to go from producing a sperm cell in the seminiferous tubules to it actually making it out of the testes through the ductus deferens as ejaculate. So it's about 2 weeks for them to finish maturing, getting that acrosome cap done correctly, making sure that we can get rid of all the cytoplasm, making sure we all have all the mitochondria and stuff lined up. Now, importantly here, these cells will get held onto for about a month. So after this 2 week time span, they will be held onto in the epididymis for about a month. If they are not released as ejaculate within that month, they will be reabsorbed, okay? This doesn't mean that we stop producing more cells. We'll constantly be producing more cells so that the cells we're producing are fresh. But those cells Like the older cells just simply get reabsorbed that because they're made out of lots and lots and lots of protein and stuff like that, lots of DNA. There's no need for us to make all new versions of all of these things. So instead, we just kind of break them down that we can use those later on. There's no buildup. It doesn't just keep building up over months and months and months and months or anything like that. After about 30 days or so, these things just get reabsorbed. So no big deal. Oh, one of the things that I didn't talk about before is that because that urethra is passing through the prostate gland, any issues that you have with your prostate gland will oftentimes be seen at the very least in issues with urination. Because the release from the urinary bladder, your bladder may hold less stuff, or it may feel like it needs to go more often just because of issues with that prostate gland. So that can actually be one of the most common it is not only one of the most common types of cancers, but this is also one of the most commonly noticed problems of someone that is having one of these cancers is going to be noticing some sort of difficulty with urination, just simply because of any issues with the prostate gland are going to have some sort of issues either with sexual intercourse or with urination. That's why it's important for older folks to get their prostate glands checked. All right, so now we're going to spend a little bit of time talking about the penis here. Now, one important thing to keep in mind is that the body of the penis is going to contain 3 particular bits of erectile tissue. Like I said before, we talked about the cavernous spongiosum, which is going to be around the urethra, but we also have 2 lateral bands of corpus cavernosus that we haven't seen before. So here we have the urethra. Here we have the corpus spongiosum, but then the corpus cavernosum, remember, is the one that's divided like into those little packets. There's one of these both on the left and on the right. Now, importantly here, in each of those cavities, or in each of those erectile tissues, we do have an artery that's going to be passing through them because we have blood that is going to be entering. And then what we're going to do is we're essentially going to have blood enter. It's going to fill up all of these extra spaces so that it can fill with fluid, kind of like putting water into a balloon. But then we're going to shut the water off, just like if you were, say, to put water into a balloon and then tie a knot on it. Now the water is stuck inside the balloon. And because the water is stuck inside the balloon with all of this connective tissue around it, that is actually going to be what is going to allow the penis to maintain its rigidity, okay? Now, because of that, though, there is no bone here, okay? There is no like hard connective tissue holding everything in place, which means this is just all a bunch of soft tissue that's being supported with the strength of fluid. Well, just like you can break a water balloon, you can break these. If you've ever heard of someone having a broken penis, it is literally because what they've done is they put enough pressure on the penis during one of these times when it's holding all this fluid that we have broken one of these erectile tissues, one or more of these erectile tissues. So whether it's a spongiosum or one of these cavernosum layers. And we have an artery, fairly good sized artery, moving through these things, which means there will be lots and lots and lots and lots of blood during one of these cases. I actually have a friend of mine that this happened to, so it is a horrific story. Hilarious, like over beers hearing about it, but a horrific story. All right. Now, all of this is to say that the whole point of all of these things is because we want to combine the genetic information from one individual with the genetic information from another. Last time we talked about how we separate everything out. We have 22 autosomes and we have the sex chromosomes. Now, genetic sex is what we're going to typically talk about as our XX or XY. However, I talked last time about how there are times when someone gets, say, 2 X's and a Y because we had some sort of non-disjunction event at the sex chromosomes. Not too dissimilar from, like, say, someone in Down syndrome getting 2 copies from one parent of chromosome 21. The same thing can happen for chromosome 23. So some people will be X naught, so they're going to get an X from one parent and nothing from the other parent. Other folks might be XXY. These are both going to be their genetic sexes. Both of these would be something that would be slightly different from what you would consider as your typical phenotypic male, female. Hello. Okay. Now, phenotypic sex is going to be what we would typically call or base off of the appearance of their internal and external genitalia, where if we see ovaries, we would call this a phenotypic female. If we see testes and male genitalia, we'd call this a phenotypic male. However, this takes a little bit, roughly 7 weeks or so, before that can be seen on something like an ultrasound. And that's because we need the hormones that are going to be secreted based off of that kind of sex determining region, or the sex determining factor, this SRY gene, basically pushing us in one of two directions. Because before this kind of 5-week period or so of development, both males and females are going to develop the same way. And they will still be developing sex organs up until this kind of 5th or 6th week, at which point they receive the signal from the sex determining region gene, and that's going to push them in one direction or the other based off of the expression of this gene and the androgens or lack thereof. So that's what we're going to see here. So 5th and 6th, we are in this sexually indifferent stage where we have basically a cloaca, like is found in lots and lots and lots of animals, whether it's reptiles, birds, stuff like that, where we have a single orifice. At which point, if we receive androgens, because we have the male SRY region, that gene gets expressed, that's when we're going to develop into the male structures here, where we start to develop the testes, ductus deferens, all those kinds of things. In females, we don't have those structures, so we would go this other way. But as you would imagine, there are some variations that can occur that would cause problems and pushing one of these to the other ways. This is actually why it can be such a problem if someone is XX or like XXY or X naught, because that can actually mean that, say someone is XXY, they will still be producing some of those male structures. However, based off of how those genes are expressed, they're actually probably not getting enough of that gene expressed, so it could actually be somewhere kind of in between here. All right, and that is basically the end of the chapter. So if you have any questions or anything like that, by all means, make sure you reach out to me and we can talk about anything that you might see. Now, functional anatomy wise, you might be wondering kinds of types of questions I ask. This is going to be your typical, like, hey, form follows function stuff. How do we know form follows function? Like, what's one good way for form to follow function in the male reproductive system? Well, we're trying to produce lots and lots of sperm. We know that we only produce sperm going from the outside of a seminiferous tubule to the inside. What do we need? That means we need lots of the outsides and lots of the insides. How do we get it? By in such a small space, because we're talking like smaller than a walnut. How do we get that in that space? Well, we just fold it up. Okay. So all of the lots and lots and lots of seminiferous tubules and all of the convolutions of those seminiferous tubules is going to be how we get enough sperm produced to be able to fulfill that 2 to 500 million that we're looking for in each ejaculate and stuff like that. So same types of questions as per usual. Nothing should be surprising at this point. But if you do have any questions about anything, if you're curious about content or testing or anything like that, just let me know. All right, thanks. Bye.